All right. Hello, everyone, and welcome to the Research Brown Bag. I'm really pleased to introduce our speaker today, one of our assistant professors, Filipa Rijo-Ferreira, which I may have said wrong. Hopefully I did okay. And she does extremely interesting work on circadian rhythms in parasitic diseases, and I look forward to hearing more about that. So thank you so much, Filipa, for agreeing to present.
Thank you. Thank you so much, Jen and Lauren, for organizing and inviting me. I will give you a little bit of an overview of what we've been doing and where we plan to go. Yes, as Jen mentioned, my name is Filipa. I am a recently joined assistant professor, and our research focuses on parasitic diseases. And I'll start with malaria, but then later on, we'll also touch a little bit on sleeping sickness, another parasitic disease. Malaria is really one of the deadliest diseases still around. And the reason it thrives so much is because the vector that transmits this mosquito Anopheles species is actually very well distributed across the globe, mostly in the equatorial regions, but also the parasite itself from this genus Plasmodium is quite resourceful, and it's a eukaryotic parasite, and these words means that it's not a bacteria or a virus, and it's closer to our own cells, and so this has been just much more challenging for the scientific community to develop vaccines against this type of pathogen. And altogether, malaria is endemic in about 97 countries, and it gives rise to about 200 million cases each year, with still about 500,000 deaths. And since this is mostly in children under the age of 5, it's completely unacceptable, even though these numbers are so much lower than what they used to be. There seems to be a plateau, and now with COVID, it's predicted that whenever we are able to resume the estimates, we will see the reality is much higher again, which is of course unfortunate. And from my own perspective, my personal reasons to be interested in these infection was actually because malaria is very famous for its periodic fevers. So even before we knew it was caused by a parasite, in ancient Greek, there are already descriptions about a disease that had fevers that would come and go. And now we know it's a consequence of the parasite replication in the blood. So this by itself, it's a quite an amazing phenomena. If you can imagine that this is a one-cell organism, this parasite, and it's coordinately invading a red blood cell where it stays, replicates, and then bursts. But all the parasites are doing so at the same time. They are bursting their red blood cells, releasing a new wave of parasites, metabolic waste, and that gives these fevers. And there are other rhythms described for this infection that I will touch a little bit later, like in the case of the mosquito biting itself. But in this replication cycle in the blood, we now know that it's also accompanied with many different beautiful rhythms in the gene expression, so the messages that these parasites are producing. So some genes are going to be up early in the day and down later at night, whereas others will come into play. And curiously, different parasite species either take 24 hours to complete the cycle, 48 or 72 hours. And this is, for us, really piqued the interest because of 24 hours or modulo, so multiple of 24 hours. It's quite an important time here on Earth, right? All of us, organisms like bacteria, fungi, plants, we're all exposed to these very predictive changes in our environment from day to night. And this gave rise to organisms to evolve a way to anticipate these changes. This is called our circadian rhythms. In the case of humans, for example, it regulates our sleep-wake cycles. So sleep itself, it's quite complex. There are many different areas in the brain regulating arousal or sleepiness, but the timing itself can be pinpointed here in this small subset of neurons in the hypothalamus called the suprachiasmatic nucleus. And this is what you can think of a master clock. So it's defining of when different physiology needs to happen in the case of sleep as well. And within these neurons, we have a molecular clock, a molecular machinery that can be simplified by different genes activating the transcription of others. And these activators, they lead to the transcription, so the expression of their own repressors, and then the repressors compete for this binding, and this itself takes 24 hours. But the activators also bind in many other genes, other their own repressors. These will be genes involved in metabolism, genes involved in immune regulation, genes involved in oxidative stress. So that's how the circadian clock really is regulating many different physiological properties of our body. And in the case of parasites, in malaria parasites, actually, this has been thought that these very well-defined human rhythms will be responsible for the rhythmic fevers. And one experiment early on, as you can see, is like from the 1930s, beautifully demonstrated how the circadian rhythm of the hosts, so in this case, they were studying in monkeys, but it can be also thought of like of humans, is extremely linked to the parasite rhythmicity in the blood. And that was because imagine that they were having a monkey and they were following their sleep, let's say. And here these wiggly forms is exactly representing when the animal, the monkey, is sleeping, and he's sleeping at night in these darker shaded areas. So the next day again, sleeps at night, the next day again. And then these researchers exposed these monkeys to a jet lag experiment. So a 12-hour light shift, like the monkey would be traveling now across the globe to a new time zone. And what happened is that this monkey experienced a jet lag, but eventually was able to resynchronize to the new light-dark cycle. What these researchers were following were actually parasites in the blood of these monkeys. And they were looking at the particular cycle that I mentioned and monitor a particular stage of the cycle. And what they found was that the particular stage was present and at certain times of the day. And just like the monkey jet lag, the parasite also experienced jet lag and then was able to resynchronize to the new light-dark cycle. So they concluded with this experiment that the parasites were being driven, their rhythmicity was being driven by the host's circadian rhythm. This could have very well been the whole story, but recent experiments that we've done, we were able to demonstrate that even parasites that are not really directly exposed to light and dark cycles because they are always inside our body or inside a mosquito, they can also have intrinsic ways to tell time. And so we thought our hypothesis that this might be more complex than just following the host, but the parasite could itself have a way to tell time. And so this was what we set out to test. And we can think of many different reasons why parasites would not necessarily want to be only depending on the host for their information of time. And that could be a way instead to be able to anticipate different changes. So think about that being in a host that eats specifically during the daytime, as us, we are diurnal, we eat during the day, we sleep at night. So the nutrient availability in the blood where the parasites are is very different from night to day. So anticipating those would be interesting for the parasites. We could think that if the parasites can also anticipate when our immune response is going to be stronger, could also be a good advantage for them. And also the mosquitoes, they do not bite at any time. They usually bite during the nighttime, the Anopheles mosquitoes. So the parasite, in order to be able to be transmitted, could be wanting to anticipate that encounter. From our perspective, why should we care? Why do we want to study whether parasites have their own clocks, have their own way to tell time? Well, I think just the knowledge of biology and the fundamental question is a worthwhile reason, but we can think of many other possibilities. So if the parasites have differences in their biology throughout the day, maybe they are going to be more susceptible to treatment at specific times. If a particular function that they need is going to be needed at a certain time, then we should treat them at that time. We can also think that if there's so many advantages for keeping an internal clock of parasites that evolved all this way in evolution, and if they still have it, these are probably going to be quite important. So if we are able to target those pharmaceutically, we are possibly disrupting their rhythms and thus their biology. So we're hoping that this could be also long-term a useful discovery. So I'm going to bring you just two slides of data of how we came up with the testing whether parasites have or not their own clocks. And these were done in mice, and we were trying to challenge the infection. So we had a normal situation in which we have a mouse group that is exposed to the normal light and dark cycles. But then we also had another group of mice that were in complete darkness. So not really exposing to the environmental light and dark that we are usually exposed. And then they would have an infection with Plasmodium, with malaria parasites, and we would take samples from the blood and assess the gene expression of the parasites. What we first analyzed was an unbiased search for rhythms. And this may seem complex and abstract, but I'll try to as well as I can walk you through this. So here, each dot will be representing where the gene expression of parasites that came from either a mouse in light darkness in the normal situation or in complete darkness looks like. And different dots that are closer together say that overall the gene expression of these parasites were more similar to each other than they are with ones that are in a different area of this plot. And what we were very excited to see was that not only what we call our biological replicates, so different mice that were collected at the same time, so A and B, so they were all collected at 16 hours, they are together, that's great, but they are also together with other samples that were collected at 40 hours and 64 hours. And if you can see all of these, what they have in common is that they are collected 24 hours apart. And that was true for all the samples. And you can also even notice that there is a circular pattern here on this graph, which means that whatever was changed and then came back to the beginning. So that was extremely exciting to us, meaning that independent of the light and dark cycles, the parasite biology was changing. And then we have some statistical algorithms that we can apply to really find through each of the raw data here in green. You can see now the lines over it. These are the algorithms feeding the data to really quantify how many genes are cycling in the parasites. And we noticed that there are about 80% of all the parasite genes that are having this up and down regulation throughout the day. And this was true independently of the light dark cycles, really concluding that these are really not important, even though the monkey experiment was playing with light and dark cycles. They are not really being able to sense and adjust independently. They are adjusting independently of light. Here you can just see the heat maps as I showed you at the beginning as well. So the many different experiments we tried, but I think the one that ultimately shows that the properties of the parasite rhythms are intrinsic to the parasite and not driven by the host, is taking advantage of a mutant mice that does not have a functioning clock. So whenever these mice are in complete darkness, so not really in a normal environment, they stop having daily rhythms of sleep, daily rhythms of feeding. And we have ways to monitor those at the behavior level. And this is the type of plot we usually represent the data. And so here is just the outcome of behavior monitoring how much the mice run on a wheel. So we have cages in which animals have access to a running wheel, and they really love to run on the wheel. So they spend they run kilometers each night. And here, this is exactly what that represents, the nighttime and the daytime. And all the black dots here is wheel revolutions. So you can see that all the activity is happening, and these are multiple days of recording at a higher representation of hours here. So you can see each night, this mouse is running on the wheel. And you can also see these red dots, and the red dots mean when the animal is taking food. And you can see that most of these is happening at the same time that they are active, similar like us, right? We are active and we eat usually at the same time. And here there's a shaded area that really is the key part of the experiment, because that's when we suddenly turn the lights out. So now these mice will not have any environmental cues to rely on, but their own circadian rhythm. And you still have a very active period and a resting period for these animals, whereas an arrhythmic mutant mouse does not. So whenever you have here in the upper part of the experiment, the light and dark, it shows that pattern of activity and rest. But as soon as they lose information from the outside world with this complete darkness, they stop being rhythmic, as you can see here. That's why we call the arrhythmic mutants. And this was really a good system for us now. Infect these mice with malaria parasites and try to see if there's still rhythms in the malaria themselves, or if they are becoming arrhythmic like the host, meaning that they really depend on host cues. And so now you've seen this plot before, and you already can tell that a circular pattern is a good sign. And that really supports our hypothesis that the rhythms are intrinsic. We were able to quantify those, and indeed, there are still 60% of all the parasite genes that are being expressed rhythmically, even in an arrhythmic host. So that really showed us very strongly that the parasite rhythmicity is not only driven by the host, but is intrinsic in its property. And you can see here that the gene expression heat maps as before, showing beautiful rhythms, even in arrhythmic mutant mice. However, there was a drop, right? Before I told you in a normal situation, there are 80% of the genes that are cycling. And now we're talking about 60% of the genes that are cycling. What happened? What was the reason for some of them to stop cycling? And there are many different reasons we can think of. It could be that they were really just responding to some rhythmic cues of the mouse, let's say to food intake being rhythmic, or it could be that similar to other circadian systems like us, and we know a lot about our cells in culture, we now are able to know that if you think about at the populational level, like we are thinking about these parasites, we are not looking at one parasite through time, we're looking at all of them, what they are doing. We could expect that when receiving cues each day, the population was going to show these beautiful big amplitude rhythms. However, in a constant condition, whenever the incoming cues disappear, the rhythms will just slower in their amplitude, and that will become just undetectable through our techniques. And maybe it's a combination of both. And so I'll show you here some of the data that gave rise to this hypothesis based on other experiments. other things we know from the mammalian system. So here what you can see is a once a day expression. So once a day, you see the light coming, and that's when we look at the brain area that I told you, the suprachiasmatic nucleus. So as a tissue, it all blinks together. But now if we dissociate these neurons and put them in culture, instead, we see that these are all blinking, each cell, each neuron, it's blinking at a different time. But if you are to follow one particular one, and you can see it here, that particular neuron is still rhythmic, is at the populational level, is that they stop being synchronized with one another. So if we would monitor now, if we would plot now all the different cells, they would all have rhythms, but not in sync. And so the overall average will be a flat line. And so we can think that, and we hypothesize that that's what's happening also to malaria parasites, that they do have rhythms, similar like the neurons. However, at the populational level, without rhythmic cues, they will lose that rhythmicity. So we tested that again, doing a new experiment that just let us look at what happens later on. And so in a normal mouse, you have here, I lost my cursor, so I will just describe it for you. On the left side, you have early on days, so day 5, day 6, day 7, and you can see that a certain stage of the parasite is either a very abundant or very low abundant, the next day again, very abundant. And that is the truth for a wild type normal mouse. Whereas in an arrhythmic mutant, we see that very beautiful rhythm at the beginning, but as time goes by, now they become asynchronous within the body, so the parasite population loses the synchronicity. And so that really seems to resemble what we know about other systems. So altogether, we also tried to test this mathematical. This was all done by John Abel, a really great, talented mathematician that was very intrigued about that and tried to recreate through differential equations different situations to test our experimental conditions. So here what I have highlighted is the model that seemed to better represent what we observe experimentally. And the key thing here is just look at the most bottom plot, is that you can see the pink lines are actually 100 lines representing each single parasite, and then the dashed line representing the populational level. And this is recreating exactly that arrhythmic mutant experiment. And mathematically, what John saw was that when you remove the rhythmic feeding, you remove the rhythmic activity of the mouse, the population of parasites shows rhythms that all through time start having a big amplitude, because the parasite population becomes asynchronous without the rhythmic cues. And so that was how he came up to the same conclusions that we did, that malaria indeed has an intrinsic clock. And this was the outcomes of its other testing of the other hypothesis. So altogether, what we've learned from this study is that malaria definitely can sense the host, but their gene expression is not being driven by the hosts, is in fact intrinsic. But this just raises now many other questions. So what are the cues that they are sensing from the host to get the population of parasites synchronous? But also, what would be the way that they regulate if this is self-sufficient? How can they do it? What's the mechanism? And the mechanism might not be so easy to find, because across different organisms, even though circadian rhythms are conserved, the molecular players, the genes that regulate it, they are not. And so these parasites will lie here, and that might prove challenge to find them, but it's just going to create opportunities, because if we are able to target them, they're not going to be targeting our own clock genes. So that's kind of the dual reason why this can be quite interesting. One thing that is shared is how important these rhythms are. And both if you disrupt clocks in plants or mammals, like in mice, but we can also represent here a human, this is really detrimental for having much accelerated aging, decreased survival, metabolic syndromes, such as higher incidences of cancer. There's so many different problems associated with disrupting that we can think also that if we disrupt now for malaria parasites, for example, these can also perturb how good they are at establishing an infection. And with that, in terms of how we are going to apply these, I'm just going to bring you very briefly some early studies we've done with sleeping sickness. And I mentioned before that sleeping sickness is another parasitic disease. It's much less distributed worldwide. It's actually only common in Africa. And that is because the way it's transmitted is through another vector, the fly, and that's endemic to the African continent. The number of cases are extremely low these days. Mostly it's believed that, unfortunately, because it's such a rural infection, it's just underreported. But there's a massive economical burden because unlike malaria parasites that are very species specific, Trypanosoma brucei, which is the one that causes sleeping sickness, is not. So there are many different animals, including cattle, that are affected. So it's about $5 billion each year worth of damage on these already, of course, vulnerable populations. So it's still quite important, and we still have to keep it in mind. And how these two come together, sleeping sickness and the circadian aspect, was because of the disease itself. This is how you would know by your own experience that us humans, we have a very consolidated sleep during the nighttime, which is represented in this plot. But see that the sleeping sickness patient, the timing of sleep is quite disrupted. And this was how we came to this interest between circadian rhythms and parasites. And indeed, the sleeping sickness parasite is able to disrupt the clock, which is another story that I don't have time to tell you today, but it is interesting how it might be doing so. But one thing we found was that the parasite itself also has rhythms in a very similar approach to how we find it in malaria parasites. But here, why I want to bring this up is because it regulates a lot of the metabolism of the parasite, so how it uses nutrients. And that is why you see here all these beautiful rhythms in the gene expression, but also at the energy level, if we look what happens, what the parasite is doing. And this was for us just opened a lot of opportunities. If we know that there's a lot of different anti-parasite drugs that act on metabolic pathways are changing through time. So perhaps the parasites are more susceptible to treatment at different times of the day. And that's exactly what this circadian medicine may mean. And so if you are to treat with an increasingly higher dose parasites, but you initiate those treatments at different times of the day, you come up with a dose response curve, which you have on the left, but you can see that there are shifts in how much dose you need to kill the same number of parasites, depending on when you started. And that's exactly what you have also summarized on the right side, that you would need 2.5-fold more drug to kill the same number of parasites if you start only 4 hours later than the earlier measurement. So this was a proof of concept how we should be considering time of day medication for infectious diseases. We are doing so, for example, for managing cholesterol or high blood pressure, that you take that, for example, in the early morning, but we have not been thinking about that for infectious disease, and there may be an opportunity. In the case of sleeping sickness, the treatment is so toxic that perhaps this small change could prevent a lot of the 5% of death that happen just as a consequence of the treatment. And so we today we talked a little bit about malaria, sleeping sickness, and about circadian rhythms. And I hope that the messages you can take is that parasites can have intrinsic clocks. They don't necessarily depend on hosts to show rhythmic biology. They still are able to sense the environment they're in, the host. And some parasites, like sleeping sickness, they are able to disrupt the clock of the host. And we could think about these moving forward for other infectious diseases, whether there's a window of opportunity of when to treat. And moving to the next, what's coming, we are definitely interested in exploring if there's this window of opportunity in terms of malaria. There are many different drugs available. Are there some of them with a very short half-life, so the amount of time they stay in the blood is very short? So is there a window of opportunity in which we should be giving the drug? And also bring this into a big perspective, including the vector in this conversation, since the vector itself also has a very rhythmic, the mosquito, a very rhythmic biting time. As I mentioned, bites through the nighttime. Did this create a pressure for an evolution that all the 3 clocks of these components need to be aligned for an infection? So just to pique the interest of where we're going, as I mentioned, these anti-malaria treatments. So for example, primaquine, we are noticing that it has a very short half-life. We realize that in the blood, if you are giving the drug in the morning or the evening, the amount of drug that you are able to find in the blood is quite different. So really suggesting that time of day drug administration might be important. All the enzymes that metabolize this drug are rhythmic on their own, so the host rhythmicity is quite important in terms of drug metabolization. So I think this is just supporting that it's worth moving forward to consider that. And in terms of this tripartite evolution, we want to test that in the lab, but we can think of how this might be important in public health context. Mosquitoes, you may have heard, because of the use of bed nets, mosquitoes' biting time is shifting a little bit from the nighttime to earlier in the day. Is it because we are pre-selecting for the mosquitoes that would naturally have that biting preference? So I'm interested in assessing how the clocks of the mosquitoes are contributing to this phenomena. We should be studying the different behavior of the populations that are being infected. Are these somehow related also to the behavior patterns? There's so very little that we know about the sleep patterns of, for example, African populations. Most of all the studies are so biased to European descendants type of either American or really from European settings. So it would be really important to expand these to other populations. And I think from the infectious disease perspective, this will be very interesting too. And just overall, how does that impact the likeliness or effectiveness that these populations have in handling the disease? So that's what I had to share with you today. Most of the work was done in my previous mentor, Joe Takahashi's lab, with John Abel, as I mentioned, also our collaborator, Maria Mota. But we have a young lab and very motivated young students that I'm happy to have in the team. And so thank you all, and I'm happy to take any questions if there are.
Great, thank you so much for that really exciting work that you're doing and sharing it with us. People want to unmute for ask questions or put them in the chat.
I'm having a hard time. Stop sharing. I lost my cursor. Okay, good.
Eva, go ahead.
Yeah, well, no, this is not really a question. It's just, I just think it's so amazing. I feel like it's like kind of the new microbiome in a way for me, because, you know, here we are plodding along with our like this immune correlate, infectious disease, that, and all of a sudden there's like this microbiome, and like it completely, it's like the gorilla in the room, right? Because it's like if you have to take into account, and it's hard to take into account, but then, okay, we're starting to understand that conceptually. And now you come in with this circadian rhythm, and it's like, oh my God, like we're doing all this totally blind. to like these really important concepts. I mean, it's so obvious when you explain it, the effect on, when, I mean, just clinically, like medicines, and also like when we do, I mean, there's so many things, and the mosquito biting, and it's just crazy how important all this is, and to figuring and how complex it is. But I just think it's so important, and like for me has really changed the way I'm thinking about things. Not that I can actually do much about it all the time, but it's just really important to put that context into it and realize how much we're not controlling for, and just it's such an important space. So thank you, Filipa.
No, thank you so much. And that's absolutely right. It's difficult to control because it's everything and in everywhere, right? So you're absolutely right. But if we can come up with different ways,
absolutely, and there's very simple truths. Yeah, no, it's amazing. Like 2.5-fold less Suriname. I mean, you would kill so much fewer people. I mean, it's like just really simple stuff, or you'd have such a better uptake. And it's just right there, like that's the tip of the iceberg, and just the tip of the iceberg could have really powerful clinical and public health, consequences in a good way.
Yes, yeah. I'm thinking a little bit about that. For example, it's not that antimalarials are not effective. There's, of course, a lot of reinfections and things like that, but there's definitely also resistance. So there's this delayed type phenotype that has been described. Can we just, boost a little bit the system by the when, right? So just sometimes it can be a small shift that is needed. It's not the all or nothing, I guess. Sorry, Jen, you also had.
Thanks, Filipa. That was such a great talk, and your work is so fascinating. I originally was going to ask about what other diseases we might suspect of having these types of rhythms, but then I also got really interested in your plot showing that the individual within one mouse, they could be following a rhythm, but because they're not synchronized, it would appear flat. And then I started wondering if like all kinds of things are cycling that we can't see. So I don't have that clear of a question, but anyway, any thoughts on that ranty observation?
No, that is actually very, very true. So just in general, of course, I think a lot about parasitic diseases, and there are many different other rhythms described. For example, filaria, parasites, they seem to be in the lymphatic system, but the microfilaria, which is a transmissible form, shows up in the blood, peripheral blood, at certain times of the day, and that actually seemed to match the time that they are likely to be transmitted because of the vector biting. So that's one that very, very likely is going to have it. We just have to now test it and find a way. Another thing is, for example, schistosoma, which is in snails. So you get it by going into infected water, and those transmissible forms, they actually emerge out of the snails also at different times of the day, depending on depending on the parasite species, whether the final host is a human or is a rodent, so a nocturnal animal, those emerge at different times. So this cannot be really a coincidence there. Most likely it's also all regulated by a circadian rhythm. It's just that there's not a lot of people thinking on that route yet. So yes, many other parasites will have, possibly beyond parasites. So we know only so far very well for cyanobacteria because of all their photosynthetic and how important light would be. But microbiome, all these other pathogenic bacteria, they may have also advantages to have at least some of their functions to happen at certain time. And we have just not gotten there because it's so difficult to go through the genetic part there, since they are not really conserved very easily between organisms. So it has to be most likely from the phenotype, so from the behavior, from the observations, and then trace it to the genetics. Your other very difficult question, whether there are many things there that are not rhythmic as a population, but might be showing rhythms. Oh yes, that's even more difficult to address. I think we would need to go follow each thing at the single cell level, which of course is very different, very difficult to do it unless you do it in a very controlled system and you have the right reporter, right, to follow. So yeah, good questions, difficult questions.
Ndola, you have a question?
Yes, thank you so much, Filipa. This is so fascinating, and I don't even do infectious diseases, but this is really fascinating. Well, my question to you is, okay, so where do we go next? How do we include this knowledge in sort of into programs, you know, in terms of control and treatment and testing, etc.? What are your suggestions?
That's good. Where should we do it? So I think at the minimum, we should be considering time for whatever intervention we do. Just keep track of when you're doing such things. So that's, I think, the basic, right? If we are also trying to implement, for example, clinical trials, there are so few that even take into consideration the time of day, either actually testing different times, but at least monitoring exactly when different people on the cohort is taking the intervention. So at least, you know, down the line, we are able to go back and try to extract that and compare if the groups had differences in the outcome because of that. So I'm thinking at right now, because these are difficult, of course, to implement. You have to duplicate the amount of things you're testing if you want to now do it twice. So if at least you are very good at controlling when each things happen, we are possibly then able to integrate. So I think that's where the most immediate, very actionable next steps are before we can take the bigger steps in the future.
Okay. So just one other follow-up question. Do you think that, for example, some of the treatments and including some of the clinical trials for malaria vaccine could be impacted by when the vaccine is given?
Yes, absolutely. I'm trying to pitch that idea to the people that are following on vaccine for malaria. So there is one study already that was testing exactly the time of day for influenza vaccine. And they noticed that if you're also an older population, so over 65, and you would receive the vaccine in the morning versus the afternoon, the morning people that received the vaccine, they had higher protection levels. So I'm thinking that can be definitely important beyond just the flu and potentially for malaria vaccines, that they are not at all as effective as we think about, for example, COVID, right? This is a 30% protection. So again, if you just do a twofold more, it's crazy how much effect that would have in the population. So I'm definitely trying to have that brought into the table also in the case of malaria.
Thank you.
For sure. No, thank you. Oh, someone, yeah.
In the chat, I assume viruses have no circadian rhythm.
Yeah, I think viruses will not have a circadian rhythm. That's right, because they have very few genes and they are not really live organisms. But one thing is already clear is that the timing at which they do things, it's very rhythmic because they are inside mammalian cells that have circadian rhythms. So just by using the machinery of the host, they will already be susceptible to the timing of the day, and that has been shown for some different viral infections, like for example hepatitis C, but others, some more recent studies have expanded that just also the timing at the immune system seems to be so rhythmic that also impacts viral infections as well. So yes, probably it's not an intrinsic property, but it's something that they will be susceptible to.
Thanks. Very interesting. Fanyong, did you have a question?
I really like the question. Maybe I'll probably ask it for question to Eva or Filipa. Okay? I think that what they call the clock. The virus may not, sense the clock, but the virus have been shown many different ways to, communicate with the cell. So they don't communicate with the organism, but they communicate with the cell. So like, for example, the cell say, I'm going to divide. The virus say, well, I don't want you to divide. Or I say, I'll go with you. And you divide it once, I will divide it once, okay? And of course, And most of the time, the virus say, I'm not going to listen to anyone because I'm going to just replicate 1,000 times even the cell only divided once. Okay, so in 99.99% chance, the case that the virus just replicate crazy, right? So, but in some of the very unusual circumstances that the virus say, I want to just hang out there, and if the cell divide, well, I have to divide, otherwise I'm going to dilute it out. Okay, so, but I don't want to replicate crazy because they're going to kill the cell. So the virus and coupled with, it's called coupled to the cell cycle, right? So they communicate with, because of our cell, the intrinsic, clock, the replication clock is that 24 hours you can replicate it once in a human cell. So the virus can say, okay, I'm going to communicate with that mechanism, cell cycle, and I'm going to replicate it once if the cell replicated once. So, I think that may be the question from Leia. Okay, again, but I feel that I want Eva and Filipa to comment on that part, because at least when I think of whether the virus have a clock, I think the virus have a clock coupling with the cell cycle, and yeah.
Eva, do you want to go?
You're muted.
Oh, wait.
No, not on that. I'm just like still kind of the immune. I mean, the whole vaccine question is so critical beyond parasites too. I mean, it has to do with the human. That's where I'm picking up from Van Young's story about. I think that's true, but I mean, the virus, it is going to be with whatever cell it's in, and it can either ignore it or then override it or stay with the cycle of the clock. But I'm just thinking about the previous question about the vaccines. It's so critical because the immune cells have their own cycle. And so it's very important from that point of view, regardless in a way of what the vaccine is too, but like
that's a great
it's
point.
so important because of where your immune cells are. And I don't think in the flu thing, I don't know that paper that well, but it seems, I mean, this is so burgeoning field, there's just like everything you turn, it's like a whole critical new question to address. And I don't think that many people, well, I guess, there tends to be like the basic scientists and then like public health, and this, I think that's what's so exciting about you, Filipa, is that you bridge this and you can say like, we're learning this, and this is like this really important, lesson for an intervention, like when Dola was going, just like you say, just notating when you're giving this, just to see whether what the effect is like 0.0, right? But like, level 1 is, can you give it at 2 different times, and et cetera. And then of course it's like, why does it have a different effect if it has a different effect, and then it goes back to the lab, and so it's just super exciting to connect the, that with the public health, aspects of it, or clinical aspects of it. I think is super exciting.
Yeah, and I think what you just said, it was a very strong, important point. Like, it doesn't even matter whether we know that the pathogens have rhythms of their own in terms of whether this could be important or not, just because we have rhythms, right? And the way we metabolize drugs is different. The way we respond to vaccines, even if it's just the adjuvant, right?
Yeah.
It's going to be different. And so the time is important independently of how the pathogen, that whatever you're targeting.
Both of them are important, but there's like they each, yeah.
Exactly. I cannot not comment. Jen, your cat is just, there's so much love in that.
Oh my God, I miss my
Speaking of
cat.
parasites.
It's so good.
She hasn't made a Zoom appearance in a while, so I thought she could, here at the end of semester, she could be back on the brown bag.
Well, thank you so much, Filipa, for sharing.
Oh, thank you. Thank you
That
so
was
much.
really groundbreaking work. I really appreciate it. And
Yay.
wish everybody a good rest of the day and a good end of the semester. So
Thank you.
everybody. Thank you.
Thank you so much, everyone.
Thank you.
Bye.
Thank you.
Filipa,
Bye-bye.
can you stay on for 1 sec, or I can call you
Sure.
separately?
Mhm.
Yeah. Okay.
Fernando is probably not,
Yeah.
he's
Okay.
probably not here. He's just pretending, and that's why we're
No, that was awesome. Filipa, so Lee and I are going to co-nominate you And we're actually meeting, we've had a meeting and we're meeting again today to like discuss it. But I'm just thinking that in the letter, you know, I mean, I can certainly use the other one as a base, or we can, but what is your project going to be? Because it would be, you know, because I'd like to bring in like and this is so exciting because I mean, and also maybe highlight how critical is it you're at the School of Public Health and you can actually, you know, bring your basic science to it. And so, but then it depends on what project it is, you know, because I mean, I can talk about in general your work, blah, 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 crosses, you know, like just what we just discussed right now, but also I could comment on, or because you do need to go in, if I remember, with a particular project, right?
That's right. So I definitely know that I'm supposed to bring a project, but I guess the good thing is that if you are able to talk beyond the project, then it's an opportunity that I'm not going to be able to do it, right? So it's basically you're adding different venues in which I can make a difference. So I guess it's not necessarily has to be through the project that I'm proposing. However, the project that I'm proposing is going to be aim kind of, it's like separated into two questions. So one is what are the malaria clock genes? So it's specifically the screen, and I think that you already I don't remember, but I think you already discussed it in the Chen Zuckerberg letter. So probably it's already there. And the other one is the part of the vector. So how does the vector come into play? How do, if mosquitoes biting time is at specific time because it's also more efficient in the mosquito biology, but also the parasites now in the salivary glands. Do they have also rhythms? And they seem to have, is it because of motility? Are they better at coming from the skin to the liver, depending on the time of the day? So that's kind of the question two, like the transmission. Question one, the clock genes. Question two, transmission. I guess that's how you could summarize it.
Yeah. You know, I've been like, oh my goodness, like searching the literature for this thing about the filaria, why they come to the skin when the black fly bites. I mean, that's, you know,
yes, you also came across that. It's cool,
I always do that in my Parasology, and there's like this 1962 paper, and there's like
always like nothing.
But like, how do they know? That's what kills. I mean, and I'm an atheist, right? But it's like, how do they know to like come, like, you know, I mean, the evolution of like that the black fly is biting at this time, and the filaria are coming to the circle. I mean, I guess it's because that's when
it's because they don't know. It's exactly, it worked, right? It's like you have an intrinsic, exactly. You have an intrinsic time, and those were the ones that were transmitted. So the ones that are around, right? The ones that were doing that, right?
Yeah, I know, but it
must be. I am totally with you.
It's not that big a deal. It's just like those are the ones that evolve, like survival of the fittest type of thing.
Totally. That's what I'm imagining, right? Some would have a way.
Yeah, because that makes sense. I mean, but you know, because I was just thinking because Isabel gave her talk yesterday, and it's kind of like, okay, this is controlled by the BMA thing, but who controls that? And like, who controls the like pituitary, that gland in the brain or whatever, the brain thing, that who's on top of that? And then who's on top of this? And then who's, you know what I mean? Like, it's like turtles all the way down. I mean, you know, it's that's
it's true. And clearly
existentially, you know, it's like, whoa, yeah.
No, but I think in the end, it must be that, you know, just some had rhythms that were aligned, some did not. And then evolutionarily, just the one that worked best for perpetuating, right? In the end is the one that stayed. But exactly, but still the question is like, how do they know it? What is the cue that they are following?
It's the cues, like, I mean, nuts.
Still, there's still a lot to learn. Yes.
Yeah, that was really cool.
Yeah.
It's fun to hear like the questions across disciplines too, you know, from that group. Yeah, cool.
Yeah, for sure. And it's just like, as I was starting, I'm like, okay, I have to remember, maybe people don't hear about RNA since forever, her, right? She's like, how?
If
Okay,
ever,
remember.
you know, like,
Yes, yes, exactly. Remember to digest. Remember.
Yeah, no, that's
So,
cool.
yeah, but
That
it
was
was fun,
great.
yeah.
Cool. So,
Cool. So yeah, that I think that, and also, did you hear that there is a follow up from the insectary? I mailed you
no, I
yesterday.
didn't. I was like, I was just
I
gonna tell
did
you.
not. I didn't email.
Yeah, maybe I'm like swamped, but let me know
No,
what
that's
happened.
good. Doesn't matter. It's
Okay,
just
is
to
there
say
anything
that
happening?
yes, exactly. Things are moving forward.
Oh, wow.
So the dean reached back, and he and Lee were behind the scenes trying to figure out. So campus didn't come forward, but the school did. So they are going to cover 80K of the difference.
stuff.
So it's
Okay.
a lot. And so it's really great. And so then I will cover probably about 35. So this is the second best option. I was hoping for the campus, but this is perfect. Done. Let's do it.
Oh, that's great. Because I was gonna
That was
say,
really
I'm
good.
on the committee of the Chancellor's Advisory Committee on Life Sciences, and we're writing this report and pushing on the like really horrendous state of a lot of the like buildings, and there's like mold, and there's, I mean, we're just trying, like, this is like ridiculous way to do science, you know. And then I was just reading, because we have 72 responses from all the life science, and there's like capital projects. Oh my God, there's like a whole list. Everyone's like, I can't recruit you, so I was like, oh my God, Filipa, you are not alone, but anyways, but it's like, oh my God,
definitely this should be a priority to change, because it affects everyone.
And everyone was like, oh no, you should tell the chancellor how fun. And my project was going to be like post-pandemic, like, yay, let's all do. And everyone was like, no, like, I can't, like, I mean, it was just like, there's such bigger problems that we were kind of like, this is so boring, but we have to be like, you guys, like, we can't, we want to be number one, and we want to recruit people, like, oh my God, like, get it together, like, and so whatever. So we're kind of focusing on this, and we got and it's like this little shop of horrors. I mean, it's like crazy, like the stories you get. So anyways, I'm dealing, like, Wendy's doing that, and then Mike Boots is doing the capital project, and I'm doing like the admin piece. I don't know if you got the survey to fill. I guess I'm not sure you were here. Anyways, whatever. So we're pulling that. In fact, I'm overdue on my piece of the report, but it's like, so that's gonna be like, because there's a new provost or something, or whatever it is, and so we're like,
and you want
like,
to
hi, welcome, like
everything is bad. You need to change these.
And you can do just these little things that'll make a big difference, or whatever. So. But anyways, good. I'm so glad. That is wonderful. There's more to celebrate. Did you fill out, like, Ashley wants to have another drinks night? Did she contact you? Yeah, we're gonna do it she wants to host the new women, and, like, our wild thing.
Awesome. No, I didn't see it.
Yeah. So she asked me, but I said, like, either next week or the week after, we're definitely gonna do it. So,
okay, great.
Yeah.
Well, that's always something exciting.
Yes,
definitely
celebrate. I'm so glad that Michael's coming through on that. That's great. Okay, good.
Yeah, no, it was good. And so I was basically thanking you guys for sending the emails. I'm sure it made a big difference, because it was also from Lee, like all Sarah's emails to him too. So thank you.
Good. I'm really glad. Something positive. Woo!
Yeah.
A lot of positive things. That's good. Okay, cool.
Thank you. Thank you for coming. Ciao, ciao.
Absolutely. That was wonderful. Thank you.
Thank you.
Okay, take care.